Please join me in welcoming to the Distinctive Voices podium, Dr. Amina Sharta.
Thank you so much for the introduction, and thank you all for being here tonight. It's a great honor to be here, especially with such a broad audience. I must admit, this is my first time speaking to such a range of ears. As Jennifer has introduced me so well, I am a professor at Scripps, and I study marine biogeochemistry. And biogeochemistry is, people sometimes think is biochemistry, but biochemistry is studying chemicals inside a cell. What we as marine biogeochemists do, we study chemicals on the planet and how they cycle through our environment. I am particularly interested in mercury, and I think that's probably because it's one of the most fascinating elements, and we have many millennia of fascination, and I always joke that mercury scientists actually invented chemistry because alchemists were playing with mercury trying to make gold with it. And so in the process of playing around with mercury to make gold, they learned about chemistry, so we invented chemistry. Anyhow, but before I jump into telling you about my research and really how mercury cycles in the environment and how climate change may possibly impact it, I had some questions for the audience just to kind of calibrate and take a temperature of what's going on here. So the questions are, we'll try to go quickly through them, so don't be shy, raise your hand if the word mercury makes you think of something harmful, like a pollutant. Okay, what about something useful? The harmful one, okay. Raise your hand if you think mercury in the ocean is primarily human-caused. How about a natural problem? Some hesitancy there, okay. It looks like human-caused, maybe next time I should ask people to scream so we can get a sound there. Okay. Who here thought about mercury levels when choosing which seafood to eat? Okay, maybe. Well, numbers are going down, dwindling, okay. I may teach you something after all. Okay, before seeing my title, so don't cheat, who here has heard that climate change might impact mercury levels in the ocean? Okay, I saw a hand over there, a few over here. Okay, that's nice. Who thinks that mercury pollution is mostly about governmental regulation? Reducing mercury pollution should be an act of government regulations. Anyone? Okay, that looks like just default, yeah, of course. How about individual action or choice? And finally, raise your hand if you've heard of methylmercury. Now keep your hand up if you know why it's dangerous for marine life or humans. I may ask you to explain, so be careful. I saw a little hand there. Okay, well that's helpful. Good, that's good to know. Okay, let's get going. So I will be trying to answer some of the questions here, and hopefully you'll walk away knowing a lot more about mercury than you ever wanted to know. So when I mention mercury, people very often think about the planet. As I was telling Jennifer earlier, sometimes I have disappointed folks because they think I'll be talking about mercury, the planet, but I'm not. And then sometimes people think about quicksilver, which is the liquid metallic form of mercury that some of you may have played with, 20, 30, 40 years ago when you broke a thermometer at home. But naturally, we have about 4 forms of mercury. And these forms, we sometimes refer to them as species. So when I talk about mercury species, I'm talking about the chemical forms of mercury. And they can broadly be split up into two broad categories. Inorganic mercury, so these are the two little balls that I drew here. And inorganic mercury is just the atom of mercury by itself, and it has either all its electrons here, which is volatile, it's called HD0 right here, or it's missing 2 electrons, and it's called HD2 because it's positively charged. The form, these two forms, that chemistry will dictate how they behave in the environment. HD0 is volatile, so that's the one we tend to emit as humans when we burn coal to make energy, to make power. Or HD2, the one that is missing the 2 electrons, because it's missing the electrons, it's going around looking for something that can give it 2 electrons, so it's just very sticky. So it'll bind to anything that has 2 electrons to give, and because of that, it will deposit. So often it sticks to dust particles, and settles and enters the environment, the aquatic environment that way. The organic forms of mercury are these two right here. And they have what we call a methyl group, so it's those little hats that I put on them. So you have the carbon and 3 hydrogens. So that's monomethylmercury. Think as mono meaning one, so like a monologue, so me here talking by myself is one, so it has 1 methyl. It's important because we'll talk about methyl and dimethyl a lot during this talk, so just keep that in mind. And then we have dimethyl, di for the Greek 2, so it has 2 methyl groups. And again, the chemistry and the behavior of those species, of those forms of mercury, is going to change depending on whether they have 1 or 2 methyl groups. So actually, these ones are the most famous ones. These are the ones that you know of without sometimes knowing that you know about them. That's because when you eat fish, almost all of the mercury is that methylated form. So when you go to a seafood restaurant and you see that little sign in California that says fish contains mercury, watch out, it's not good for you, what they actually mean is fish contains monomethylmercury. Be careful. And so what we do is studying how do we go from humans releasing a bunch of these inorganic species into humans eating a bunch of methylated species when they consume fish. So how does this happen? Since the discovery of monomethylmercury, it's the one with the 1 methyl group, researchers have been trying to figure out what is the pathway of formation. So this started around the late 60s by 2 Swedish scientists. And over time, we painted a picture, which I kind of simplified here for you. We are saying, okay, so that inorganic mercury that has no methyl group gets converted into this monomethylmercury by some microbes in the ocean, or it can happen in the sediment over here. And then it's being taken up by phytoplankton, which is, the algae that lives in the ocean, which kind of serves as the base of the food web, quite a bit. So the algae suck up so much mercury, methylmercury, that the concentration can be 10 times, 100 times higher than what we measure in seawater. And then through a mechanism called biomagnification, the methylmercury increases substantially as you go higher and higher in the food chain. And this is one of the characteristics of monomethylmercury, and that's driven by the fact that it enters a cell really easily. So it can go through a membrane, goes into the cell, but it's very difficult for a body to eliminate the methylmercury. So once it goes in, very little of it comes out. So if you're something that eats something else that eats something else, you'll be exposed to more and more and more methylmercury over time. And if you're a shark and you're sitting on top of a food chain, you're mostly all methylated mercury. That's why they say don't eat shark. So another way to illustrate the same thing with some data here, this I took from this paper. It shows how on the right here you have these reservoirs like atmosphere, and on the left here you have marine mammals. And you can see the different species of mercury. That purple and blue are those inorganic forms. That dark blue is monomethylmercury. And as you can see, as you move away from those abiotic, non-living reservoirs into the living organism, almost all of it is methylated mercury. So the question that we try to answer is, how do we go from the left here? Is that the left? No, that's the right. The right here, PhD doesn't know right from left, to the left here where we have mostly methylated mercury. Unfortunately for us, or fortunately, I guess depends on how you see things, that means we have to study mercury across multiple research disciplines. We can't just do chemistry, we can't just do biology, we can't just do health, we have to do all of the above. And that's because mercury gets emitted through a range of human activities. So from, like I said, coal burning, mercury is used in artisanal gold mining because it forms an amalgam with gold or silver. Once you burn that amalgam, all the mercury evaporates because it's volatile, and you're left with pure gold. So it's very useful. It's not very dangerous, too, but people love it because it's cheap and easy, and you can do it without having to need a factory. Once it's emitted into the atmosphere, it deposits. So you have to think about the atmospheric chemistry reactions that results in converting that volatile mercury, Hg0, into that sticky mercury, which is Hg2. You need to think about precipitation patterns. How much rain is a region getting? Is there a lot of dust that is going to settle that mercury? So these are the things we have to think about. Once that inorganic mercury settles into aquatic systems, microbes intervene, and they start picking it up, and they start transforming it. So which microbes are doing it? Under what conditions? Where? Is it in the water, in the soil, in the sediments, etc.? So these are the questions we need to answer. And then, once that methylmercury is formed, it's being taken up by algae. Well, does all phytoplankton take it the same way? I don't know. Does it matter if you're in a temperate region, in a polar region? So these are some of the other questions. And of course, we talked about the food web structure. Who eats the phytoplankton that contains the mercury? Do they eat a lot of that phytoplankton? Do they eat not a lot of it? Who eats that prey? And then finally, on the human side, well, we choose which fish we fish and what fish we eat and what seafood we consume. This is based on industry. This is based on trends. What's the latest thing on Instagram or TikTok? What kind of seafood you like is also very cultural. Where are you consuming that? So this will impact, again, people's exposure to that mercury because they'll make those choices. So these are things that we need to also understand. So today what I'm going to try to do is kind of give you a quick tour of a few examples of the research we're doing on the different aspect of that mercury cycle. So there has been a concerted effort globally in the past decade or so to reduce the amount of mercury that we emit into the environment by changing our industrial habits. Mercury is a very useful element. It has a lot of chemical and physical characteristics that are very desirable. And so it's used for a bunch of things that we bought this from Amazon just a couple days ago. It was delivered and they were saying it's mercury free. I was like, well, cool, but that means that some of the batteries are not, right? So there was mercury being used to make just simple 9-volt batteries for a while. And so the ultimate goal of these global efforts to reduce mercury emission is that we want to reduce human and wildlife exposure to mercury, which for billions of people comes primarily from not from direct occupational exposure, aka breaking a thermometer and playing with the mercury, but comes from the consumption of seafood. So on the underlying question that is guiding many of us is, if we reduce emissions, are we going to see reduction in the oceans? And if we're seeing reduction in the oceans, are we going to then see reductions in the fish, right? We are, of course, hoping that the answers to our questions are yes, but if the answer is no or not really, then what else can we do? We'll start with thinking about our first question, and let's look at emissions over time. And so this paper by Arya Fanberg just recently came out a couple weeks ago. Coming with some good news is that you don't really need to understand what's going on. The only thing you need to know is that on the left here, you have older collections of data, and then on the right here is like today, right? So time goes from left to right. And so what we are seeing here is mercury concentration, and the trends are going down. So what we are seeing is that in many monitoring stations around the globe that are measuring mercury in the atmosphere, the lines are all going down, with one exception, but that's just one station. But almost everywhere, they're going down. So it looks like the emissions are declining globally, which is a good thing, right? But the question then is, if the emissions are going down, are we going to see lower mercury levels in seawater? And the other complication challenge that we have to deal with in the research community is that climate change is happening at the same time. And so how is climate change going to potentially counteract or help reduce the emissions is something that we need to figure out as we work through this. So to answer this question, or at least try to answer the question, we'll go to the Arctic, where the concentration of mercury in the biota are particularly elevated. And the climate impact are some of the most, the greatest that we see on the planet. As you've all heard, the sea ice is melting, permafrost is thawing, things are changing really fast in the Arctic. And so if we are going to understand how climate may impact mercury cycle somewhere, the Arctic is probably one of the best places for us to go look into. So we did some work there that was requested by the Arctic Council through their AMAP program, which is the Arctic Monitoring Program. And they were interested for us to answer the question, what will the Arctic Ocean mercury look like under future scenarios? And they wanted us to look at both the mercury emissions response and also the climate. So to do this, we developed a model, kind of simplified it here for you. And the model kind of divides the Arctic Ocean into different boxes, and we kind of like sprinkle a little mercury in it and see how it changes over time. And we also try to look at how a set of environmental and climatic variables may impact the mercury levels. So we tested several policy and their implementation. So what we are looking at is we rebuild the historic changes in mercury deposition in the Arctic, and then we make some predictions based on different policy action. One is we're not going to do anything, we're going to continue business as usual, having fun, right? Just keep it burning it. And then we have some policy action, what happens if we reduce mercury emissions because we stop burning coal and we now use solar panels, for example, right? Or we only use wind turbines in like half the countries. And so those policy action will have an impact on mercury emissions. We also looked at which type of climate variables are going to have an impact potentially on the mercury cycle. And we know that in the Arctic, we see large reduction in sea ice coverage. So we expect that by 2050, we'll see about 20% reduction in sea ice coverage. We know that the river discharge is increasing, so all the rivers are draining more water into the Arctic Ocean. And once you remove the sea ice, there's more sunlight. Once you bring more nutrients from the rivers, you're going to grow more phytoplankton. You're going to see an increase in primary production. That is potentially going to have an impact on mercury in the Arctic. So we looked at how these policy actions are going to result in a change the mercury concentration in the seawater, and we also looked at how the climate variables are going to do that. And so, long story short, let me just show you, like, this side is bad, and that side is good. So you want to be on this side. You don't want to be on this side. So the good news here is that if we do nothing, it's bad. Simple. If we do something, anything, doesn't matter what it is, it's good. However, we see differences in terms of how aggressive we are in terms of reducing our emission, and also when are we implementing those. So if we do something now, even if it's small, it's better if we wait than if we wait 20 years and do something more aggressive, right? So because the mercury that we emit, just like the CO2 that we emit, builds up in the environment. So if you wait too long, then the effort that you need to make to reduce it is going to be even greater than if you did something today. So this is a conundrum that we have for a lot of the chemicals that we're emitting into the environment that are long-lasting. What the AMAP Council wanted us to look at is also answer the question, well, what is the climate going to do? Well, Mercury is volatile. So if you remove the sea ice, maybe all of it is going to evade from the ocean and everything is going to be great, right? So we're like, okay, let's look into this. So we reduce the sea ice, as we said, about 20%, which is what I expected by 2050, and we look at what it does to mercury, and we see that indeed, yes, we see a reduction in mercury if we remove the sea ice. The rivers will bring more mercury in because they're just draining a bunch of soils, okay? They'll bring in net primary production kind of wash. Here, overall, we see a slight reduction due to these climate variables, but the good news here somewhat is that that is smaller than the impact that we can make through policy action. So while climate change can have an impact on the mercury level in the ocean, that impact is not as great as us deciding to do something. And so we are better off choosing to do something than waiting for climate change to, I guess, help us. Okay. So just a reminder that this side is not good. But, so as you see, most of what I talked about is on the inorganic side of mercury. So we said, okay, we're seeing reduction in emissions. We see that this will possibly reduce the mercury levels in the ocean, so we're pretty happy about this. But it doesn't tell us anything about the organic form of mercury. That is the one that we're really concerned about because that's the one that ends up in the fish. So we will then go on to talk about our second chapter about mercury methylation and what drives it. So natural methylation, I mentioned this a few seconds ago, well, a few minutes now. Natural methylation is microbial, and it's been shown for the first time in this 1969 paper by those Swedish researchers. And they, you will note here, interestingly, they posited that maybe living organisms have the capacity to methylate mercury, and they said that they think that they found both mono and dimethylmercury production in the sediment and in rotten fish. So that was their big conclusion. Fast forward to 1990, so this paper came out by Rob Mason and Fitzgerald, both the University of Connecticut now. They found that actually, let me just show you what they found. They found that there's a bunch of methylated mercury, and I think this is the first paper showing that in seawater, it was Pacific Ocean. So here, that was only in the sediment, it was an experimental setup. They went out into the ocean in late 80s and found a bunch of dimethyl, which is that black line here is dimethyl, and some monomethylmercury in the water column of the Arctic Ocean. And one thing that was also interesting is that they see that increase where the oxygen levels are low in the ocean. So you would think seeing this, then everybody would then rush to try to understand what is that dimethylmercury species that we clearly find a bunch of it in seawater, and how does it relate to monomethylmercury that we find in the fish? But that didn't happen. It didn't happen at all. And the reason it didn't happen were twofold. One reason, and I'm telling you that, but I don't know if that's true. One reason is monomethylmercury is the form of mercury that is found in the fish, and so people were just like, oh, we need to figure out monomethylmercury. And the second reason, and I think that's the more likely reason, is that shortly after that Mason paper came out in 1990, Karen Wetterhan dropped a few drops of dimethylmercury on her latex gloves that crossed that gloves, and she passed from dimethylmercury poisoning shortly after. And that has created a lot of things and changed a lot of things in the way we do lab work. And EHS rules have changed in the US and all over the world. We switched from using a latex glove to using nitrile gloves. And the research that required manipulating or touching or working with dimethylmercury just pretty much stopped. People continued to measure it. So what people did is that they still go to the ocean and they take some water and they measure the dimethyl that is present in the water, but they don't actually work with the dimethyl itself in the lab context to understand its formation, decomposition, and whatnot. This is a paper that it was looking at transect in the Pacific. So this is depth profile, and so this is pretty much distance. And here on the right, you have the concentration. And I put my little monomethyl and dimethylmercury here. So this is monomethyl, this is dimethyl, and you'll notice, again, we have a lot more dimethyl. So red means more, and orange means more than monomethylmercury. And they're particularly elevated in those deeper, around, you can see this is 3,000 to 4,000 meters. It's pretty a high concentration at depth. And again, so this raises again the question, where is that dimethylmercury coming from? What is this relationship with monomethylmercury? Why do we not know anything about it when it clearly is in some water masses, the majority of the organic mercury that people care so much about? So in 2020, I recruited a PhD student. His name is Hannah Adams, right here. And she was equally intrigued by dimethylmercury as I was. And we decided to set up an experiment where we were trying to figure out, what happens to that dimethylmercury, what becomes of it. And so we did that in the California coastal region. And what we did is that many of you must know that this is an upwelling region, so we have these deeper waters that I was showing you earlier coming up to the surface, and they bring a bunch of nutrients, and phytoplankton loves it, so there we have those blooms, and then that water just moves away, and then there's more coming. And so this is an important region because, as you heard, monomethylmercury is bioaccumulative, so when you end up having a lot of biological activity and productivity, then you're more likely to take up that monomethylmercury if you have a bunch of phytoplankton being formed. So we wanted to know that dimethylmercury that may be upwelled there too, what happens to it? So she followed this parcel of water at sea and looked at what happens to the dimethylmercury in the water. And then she used a model to test different hypotheses. So what she found was that as she followed around that parcel of water, the dimethylmercury was breaking down, so it was disappearing, but the monomethylmercury was staying pretty much constant. And so we hypothesized that was probably because that dimethylmercury was breaking down into monomethylmercury along the way that allowed the monomethylmercury concentration to stay constant. So she used then her model to test various possibilities and whether that's possible. And so her big conclusion was that indeed, when you have this upwelling coming up, you will produce quite a bit of monomethylmercury just by breaking down with sunlight that dimethylmercury that keeps getting upwelled. And that this monomethylmercury production from dimethylmercury dwarfs the other forms of monomethylmercury production that we know of. So dimethylmercury could be an important source of that monomethylmercury that everybody cares about and potentially have an impact on seafood mercury levels. Okay, so now that we know that monomethylmercury can be produced from dimethylmercury, we need to understand where is that dimethylmercury coming from. Like, we see that there's a bunch of it there, and we also know that he can produce that monomethylmercury that biomagnifies. So, but we stopped working with it 30 years ago, so we have no real way of being able to answer those questions. A few years ago, again, a Swedish brave scientist named Sophie Johnson, somewhere here in 2010s, I think, decided that she actually wants to start working with dimethylmercury again. And so she came up with a way to make dimethylmercury safely using just much lower concentration of inorganic mercury during our synthesis process. So we just start with really low concentration. So when we make our standards, they end up being almost as low as seawater. So they're not dangerous. So she recruited a PhD student named Johannes West, and together they spent some time figuring out degradation mechanisms for dimethylmercury. And when Johannes graduated, he joined my lab, and we started 2 years ago actually making dimethylmercury here in our lab and using it to test both the formation and stability of dimethylmercury in seawater. And we also worked really hard on figuring out whether we are measuring it properly in seawater or not. Because if you just go out to sea and you measure something, but you have no way of making sure that you are actually measuring what you think you're measuring, or you haven't tested your methods with the actual elements to make sure that when you collect the water sample, that the element doesn't break down by the time you bring it to the lab, then you don't really know what you're measuring, right? There's no way to check. So if we make dimethylmercury in the lab, we know its concentration, we can start experimenting with different preservation techniques and analytical techniques and making sure that we actually measure what we think we're measuring. So we spent a few years doing that, and I think that because of that work, we have tested our entire analytical process just to make sure that we can measure dimethylmercury properly. And dimethylmercury is highly volatile, so it's really easy to lose it as you are manipulating it. So using our new approach, we measured dimethylmercury concentrations in coastal areas that are substantially higher than many other reports of dimethylmercury concentration around the globe that we illustrated here. So, okay, so we see that we have a bunch of dimethylmercury in the coastal area. We know that it breaks down into monomethylmercury, but we still don't know where it's coming from. How is it formed? So to do that, we are very lucky that mercury has 7 stable isotopes. This is kind of one thing that makes us a very happy bunch of scientists. Isotopes, for those of you who don't know, are just elements of mercury that have a slightly different mass. So they're all mercury, but they don't have the same mass. So our mass specs can tell those different atoms of mercury apart. Some of them are heavier than others. And so what we do is that we take all of these, so we purchase these different mercury isotopes, and we synthesize our own organic mercury species in the lab. So we can make different forms of dimethylmercury that are easily labeled and separated by our instrumentation. And then we can put them all in our bottles, and we can incubate them over known amounts of time, and then we can measure the different forms. So, for example, we can have isotopically labeled dimethylmercury that we let sit in the water, and then we look at how much of, let's say this one is marked 198, how much of the inorganic that is formed is 198. So we can make all these distinctions within the same bottles and look at how the different species convert into each other. If they all had the same mass, we wouldn't be able to tell them apart. So looking at the sample, you can't really tell. But because we can assign them a different isotopic mass, we can say, oh, that monomethylmercury came from the degradation of that dimethylmercury, or this dimethylmercury was formed from the degradation of this form of inorganic mercury. And from there, we've managed to answer many questions. I'm not going to go through all of them here because we don't have time. This is about when I start to panic in terms of time. So dimethylmercury production here, we used those labeled isotopes to look at how it's formed. So we did manage to demonstrate that we can form dimethylmercury in seawater over time. And one thing that we managed to show too is that that formation is biological. So this is just one example of how we did that. We incubated some filtered waters, we incubated some unfiltered waters, so we remove a bunch of particles, and then we look at how that impacts the formation. As you can see in the filtered waters, we see no formation of dimethylmercury. So whatever is doing the formation has to be some sort of particles or a microbe. We did some temperature tests, so we are toward leaning that it's not a particle, that's probably microbial. And we found, for example, that dimethylmercury formation is temperature sensitive, and it's probably microbial. So what are the implications of these findings for the mercury cycle? If we know that they're temperature sensitive and we know that global temperatures are changing, and if we know that it's microbial, we need to know which microbes is doing that and how are they distributed around the globe? Are they moving around? So the next step, now that we have a non-answer for this one, the non-answer here is that we still don't know. This is an active area of research. In the past 2 years, we've managed to measure dimethylmercury, figure out that it can break down into monomethylmercury. We think that is biological, and so we are now in the process of trying to figure out like who is doing that work. And without understanding the relationship between monomethylmercury and dimethylmercury, we can't really tell how it's going to respond to climate change. But we can still skip that part and really focus on the next step, which is biomagnification. This is something we know for sure. We know that once that monomethylmercury is present in the seawater, it will enter the food chain. And a little bit of background here is that we need to understand bioaccumulation and biomagnification and what they mean in order to understand how they may impact mercury accumulation in the seafood. So bioaccumulation comes from the bioenergetics of an animal. So it's the process by which the animal consumes energy from other animals, usually or plants, and that energy gets distributed for its growth, reproduction, waste generation, et cetera, et cetera. And so in the process of consuming that energy, the animal also takes in mercury. So the more energetic you are, the more energy you consume, the more mercury you will also consume, right? You will take up. So if somebody eats like a pound of sardines every day to sustain their energy, they will take on a lot more methylmercury than something that eats half a pound of sardines. Kind of get the idea here. And so as the animal grows, and I said that earlier, the methylmercury enters the body, struggles to leave the body. That's driven by the energetics of that animal. So over time, as it ages, it will accumulate more and more and more mercury. And then we have the food web interaction. What does this animal choose to eat as it ages and grows? And these food web interactions are what drives the biomagnification process of methylmercury. So what this animal eats is going to determine where it is on the pyramid, and it's going to determine how much mercury it's going to accumulate. These processes are highly sensitive to climate for multiple reasons. The bioenergetics, a lot of these animals are cold-blooded, so their energy level is going to be linked to how hot or cold it is. The food web interaction depends on what prey is available, and the prey may move depending on the environmental conditions. A lot of animals have very narrow temperature sensitivity, so if they don't like how it feels over here, they're going to move somewhere else. And if you're a predator who can't move for whatever reason, you will have to choose to eat something else, and that will in turn impact how much mercury you take up. So we can test this by developing models of food web interaction and bioenergetics. So this is just an example for you looking at how when, I think it's a dogfish here, grows, how much mercury it accumulates based on bioenergetics and what it eats. So this is kind of our base model, so you can see our dogfish mercury levels go up as it gets heavier. And then we can say, hey, we're going to change up the diet of that dogfish. In our model, instead of feeding it what it was eating in 2000, we're going to be like, oh, we're going to find some numbers about what dogfish was eating in the 70s and see what it means for mercury. And as you can see, the 70s diet and that base 2000 diet are different in terms of the amount of mercury that they take in. What happens if you heat up the water in our virtual aquarium. If you heat up the water, the bioenergetics of that animal are going to change a little bit. So you're going to see a slight increase in the mercury that they accumulate over that same growth period. Well, what happens is that these things can propagate in a food chain. So if you are sitting on top of the food chain, the results of these changes will be compounded. So let's say that this layer here is highly sensitive to temperature, or that what they eat, their favorite prey, this moves and another prey shows up that has more mercury in it. These ones are all going to accumulate more mercury. So if you're sitting on top and you're a bluefin tuna and you're consuming all of these things, you will accumulate more mercury as a result. And so doing this, we saw, for example, that we can see changes in methylmercury over time in tuna that are driven solely based on temperature, that are driving the bioenergetics across that whole food chain. So all the animals that are underneath that bluefin tuna that have slight increase in activity level because of slightly increased temperature, they end up all of them accumulating a little bit more mercury. And so if you're a bluefin tuna and you're sitting on top of that food chain, your response will be you'll have higher mercury levels because the temperature is slightly higher. What does this mean for humans? Well, we go out the ocean and we fish that fish. And in the process of fishing that fish, we fish the methylmercury. So this paper actually also just came out a few weeks ago where they looked at the global distribution of fisheries, large fisheries, and also the concentration of mercury in those fish. And they built this map of what they call the methylmercury fish. So it's how much methylmercury we extract from the ocean when we collect that fish. And as you can see, the distributions are highly varied around the planet. And what is going to happen is that if the fish moves because of temperature preferences, and the fisheries have to move to follow that fish, depending on where the fish is going to move, we are going to see a different amount of methylmercury being fished out of the ecosystem. So this is something that we currently are trying to figure out in some of our work. And finally, I got just a few minutes to finish up. Okay, what does it mean for humans? The amount of mercury that we consume through fish is pretty well correlated with our levels in our bodies. And we can do this by asking people how much they eat of what, the size of their meals, and we can measure the mercury levels in that food that they eat. And so we did this study back in 2016 where we modeled what people's concentration of mercury in their bodies will be based on how much they told us they ate and what the concentration are inside that fish that they ate. And then we compare it to the hair mercury that we measure. And so that's the one-to-one line. There's generally a pretty good relationship between what people say they eat, how much mercury is in that food, and what is their own body burden. So what it means, in the way it's positive, is that if we can reduce the mercury levels in the seafood, we can also reduce people's exposure to methylmercury, which is a well-known neurotoxin, and it's not good for people. We like to think about what it means in terms of climate change. How does climate change potentially impacting methylmercury levels in the seafood and what it would mean for people's exposure? We don't truly have the answers right now, but we recently received some funding to support some of that research, and that work is going to be done by Pepe Wu, who is a recent postdoc who started in my lab, and she is supported through NIEHS and NSF. And so kind of more or less a concluding slide about some of our answers. Will mercury levels in the ocean decrease over time? Probably. Looks like they're already doing that. Question 2, can emission reduction lower the levels in the organic mercury in ocean and fish? Can't really tell until we figure out this dimethylmercury question. Is there something else we can do? Maybe. Let's say if none of this works and we can't see is there anything else we can do? And this is some kind of sneak peek of some of the work we recently started, is to see if we can harness the human gut microbiome to reduce methylmercury burden on humans when they consume fish. And so we've been working with collaborators at UCLA to develop intervention methods in the human gut to actually reduce methylmercury from consumption of seafood. And we've been having some pretty good luck so far with that, so we're really excited about potential approaches if all else fails. And on this, I would like you to thank you for your attention, and go ahead.
Thank you very much.